Well, I'm very honored to have this chance to be the sort of last person to speak, last man standing, I guess. So in fact, this is a very interesting topic, and it's one that we could have spent the entire series on and still have just scraped the surface of this topic. So what I'm hoping to do is in fact touch on some very key and interesting ideas when it comes to our thoughts on how HIV causes AIDS, how it in fact co-opts very key cellular processes, and how it actually uses the immune system against itself to result in immune collapse that ultimately drives the actual formation of the disease itself. But what I'd also like to do is to make this more of a free-form sort of presentation, because it's such an interesting and varied topic that many questions will come up. And so we have definitely built in time for you to ask me questions, because there's a lot of directions that we can go with this talk. So please keep in mind that you have absolute freedom to raise your hand and stop me and ask a question, because I'm absolutely sure that any question you could possibly ask me will probably be a very interesting avenue for us to talk about this evening. Now, of course, this is a very auspicious and appropriate day to have this talk, because the 1st of December is World AIDS Day. And so every year, the 1st of December is identified as World AIDS Day, and it's an opportunity for folks around the world to really refocus their attention on the epidemic and really think about where we stand today as far as this very serious epidemic is concerned. Each year, there is also a particular emphasis on a specific issue that we think is very important in terms of understanding where the disease stands today. And so as you can see from the banner, in 2004 today, the focus is on women, girls, HIV, and AIDS. And in some ways, this underscores a very dramatic change in our understanding of the disease and our view of the epidemic that has been gradually occurring over the last 5 to 10 years, but which perhaps the public is not as aware of as it should be, which is namely, as you know, when AIDS first appeared in the late 1970s going into the early 1980s, it was very much considered to be a gay disease. And at the same time, it was very much considered to be a disease primarily of men. Now, by 1982, it was quite clear that both men and women were equally susceptible to infection by HIV. What many folks don't realize is that today, when you think about the global numbers of individuals that are infected with the virus, close to 50% are either women or girls. So at this point, 47% of the total that are infected globally are either women or girls. What is also important to know is that that gender is the fastest growing group of new infections. So that in fact, if you look at East Asia, and if you look at, for example, the former Soviet Union, the former Soviet bloc countries, what you'll see is that in the past 2 years, the number of women that have been infected by HIV has jumped by 56% in the last 2 years. So this banner and the focus of World AIDS Day in 2004 is really an attempt to get the public aware of how seriously at risk women are globally from this disease. So what I'd like to do, though, prior to really talking about the virus, which is of course what we see right here, and in fact what you're looking at is a scanning electron micrograph of the surface of a CD4 cell, and these little globules that you see right there are in fact new virions of HIV that are budding from this infected cell. Prior to us really talking about sort of what this remarkably pernicious virus can do, what I'd like to do is to give you sort of a broad overview of where we stand today as far as the epidemic is concerned. And so what I'll do is I'll start off with where we stand today with HIV and AIDS in the United States. Now, as some of you may be aware, in the late 1990s, Time magazine had a cover that posed the question, has AIDS been cured? And really what drove that really striking cover was the fact that from 1996 to 1997, the number of AIDS deaths in the US decreased by 42%. And this dramatic decrease was really due to what remains the current gold standard in AIDS therapy, which is what's called combination therapy. So this is in fact a combination of 3 different antiretroviral drugs. And in the late 1990s, it was shown that that combination of drugs could actually inhibit the replication of the virus to such a degree that you could barely detect it in the bloodstream in many patients. And the thought was perhaps we had the disease licked, that possibly that combination of drugs could make AIDS analogous to something like diabetes. Still very serious, still somewhat life-threatening, but something that you could control for a very significant period of time. So that in 1997 it was thought, aha, perhaps we had found a drug combination that could make this terrible and otherwise lethal disease a livable syndrome. Unfortunately, by the year 2001, it became quite clear that combination therapy, which by 1997 hadn't been present in any patient or used on any patient for longer than 2 years, by 2001 it was clear that by roughly 4 to 5 years, combination therapy fails, like every other therapy that we currently have to treat HIV. The reason being that even with that complex mixture of drugs, the virus is so plastic and the mutation rate is so high that you still see the emergence of resistant strains. So unfortunately, the question that was posed by Time magazine in 1997 has of course been definitively answered by the year 2001. Unfortunately, AIDS has not been cured. And at this point, there is little strong evidence that we're close to curing this disease. But as far as the epidemic is concerned in the US, currently, and the latest numbers we have are as of last year, there are over 1 million men, women, and children that are living with HIV in the United States. In that year alone, there were over 50,000 new infections that were recorded. And in fact, in the US, if you look at total numbers, there's been a stable number of new infections of approximately 50 to 60,000 per year. What's interesting is that of these new diagnoses, at this point, almost a third of them are accounted for in women. So that women are a fast-growing group in the United States that are being infected with this virus. Question?
So what's the gender ratio if you don't account for just the new ones? What's the gender ratio of infection currently?
Okay.
The new ones?
Right, so globally the gender ratio is half and half. In the US, we're looking, I believe, at 18% women. Yeah. So in 2003, almost a third of the new diagnoses were in women. But what is interesting is that if you start to break down that total by race and ethnicity, you see an entirely different picture. In that what you will find is that among new AIDS cases in men, 61% were in African Americans and Hispanics. So more than half were in African Americans and Hispanics. Among new AIDS cases in women, 80% were in African Americans and Hispanics. So what that means is that African American and Hispanic women are at an extraordinarily high risk for contracting this virus in the United States. And in fact, as of 2003, AIDS is now the #1 killer of African American women in the age group from 25 to 34. So that even though things in terms of total numbers in the US look somewhat stable, when you break it down by race and ethnicity, quite clearly among African Americans in particular, the epidemic is galloping out of control still, and that is especially true in African American women. What is also unfortunate is that as of 2002, HIV incidence among African Americans is now 8 times higher than what you see in Caucasian Americans. So what this means is that even in the United States, where, if you will, the effort to push the boundaries of AIDS education is perhaps the strongest in the world, you nevertheless see a very serious epidemic that is not showing any signs of abating. Now, if we move to the global realm, we see that the situation is in fact truly dire. And you're probably aware of this, because this has been discussed in the news fairly extensively. For one thing, HIV infection is significantly more common than we first thought, perhaps 5 years ago, and greater than 40 million individuals globally are now infected with HIV, and that's a conservative estimate. In 2003, roughly 5 million were infected at a rate of approximately 14,000 new infections each and every day. Of these 14,000 new infections, 2,000 are in children younger than 15 years of age. So once again, this is a demographic that is very striking. 12,000, the other 12,000 are primarily in individuals in the age range of 15 to 49 years old. So what is very striking about this is that, unsurprisingly, this epidemic is striking the core of the population that essentially the economy of any society rests squarely upon. And so in a few minutes, I'll talk a little bit about what's happening in Africa in terms of anecdotes. You'll see that this has very serious consequences for the economy and, in fact, the social fabric of society. At this point of new infections, roughly half are in women, and half of the total are, in fact, in the 15 to 24-year-old age range. So truly, that segment of the population that is most responsible, if you will, for the working population and for the forward movement of society. Conservatively, it's now estimated that globally, roughly 1% of all sexually active adults in the world are infected. And based on current projections, 90% of the infected don't know that they're infected. So in other words, doing a projection based on sort of random screening at health clinics, and of course polling of individuals in terms of whether or not they had any sort of awareness that they might be infected, only 1 out of 10 had any inkling that they might possibly be infected. Of course, because this is a sexually transmitted virus, this has grave ramifications for the epidemic. In terms of the death toll, at least 3 million died from AIDS in 2003. Of those that died, more than a third were adult women, and a fifth were under 15 years of age. So what we can see is that even though the epidemic is far from under control in the United States, globally it is actually burning like a wildfire that's entirely out of control. And many have argued, I think very convincingly, that this disease, this epidemic, remains the gravest challenge in terms of public health that humanity has ever faced. And of course, what is interesting when you think about this is that despite the seriousness of this epidemic, this is still a disease that can be prevented. It is not a virus that is passed, for example, through the air. It is not like influenza. This is a disease where the majority of these infections, greater than 75 to 80%, are sexually transmitted. And so it's a very interesting statement of human behavior and, in fact, social structure, that nevertheless, the epidemic is still so entirely out of control globally.
So globally, would you say that so it's a mixed bag of no education and drugs are not accessible to most people throughout the world? Is it a combination, or is it mostly because of the plasticity?
I think, so the question was, why is it so out of control? Is it primarily due to the mutation rate? Is it due to a lack of education? Or is it due to a lack of drug availability or therapeutic availability? It's somewhat a combination of all three, but I would say primarily a lack of education is a very important issue, but also the social construction of many of the societies that are being hardest hit by HIV is a very key factor. And I think with some of the anecdotes that I'll share with you in a few minutes, you'll see in particular why this is a problem. So obviously from these numbers, we are in a very grave situation globally. But I think to really grasp how serious this might be, we need to take a look at the continent that is hardest hit by HIV, which of course is sub-Saharan Africa. And so what we can see with Africa is that if you look at causes of death. If you look, for example, at lung cancer, tuberculosis versus AIDS, and if you look at global percent in blue versus the percent in Africa, as you can see in lung cancer, generally as a cause of death, it's higher globally overall than what you'd see in Africa. In tuberculosis, it's roughly the same in terms of the global total versus what you'd see in Africa. If you look at HIV and AIDS, what you can see is that at this point, it's over a fifth of deaths in Africa are due to HIV and AIDS versus roughly 4.2% globally. So that what you're seeing is that as far as the death rate is concerned, on that continent, HIV and AIDS is having a very dramatic impact. Sort of another way to take a look at this is the impact it's had on life expectancy in a variety of countries in sub-Saharan Africa, in particular in East Africa and in South Africa. And what you can see is that, and this is in fact a very sad figure, is that if you look at countries like Botswana, for example, due to improvements in healthcare, you go from a life expectancy of less than 35 years in 1955 to a life expectancy of almost 60 years by the mid-1980s. With the emergence of HIV in Africa from the mid-1980s on, what you can see is that it has returned the life expectancy of many of these countries back to what you would have seen in the 1960s, early 1970s. So talk about a dramatic reversal of progress in terms of overall health and life expectancy in these countries. Clearly, HIV and AIDS has had a remarkably significant impact. And in fact, in some areas like Swaziland, it's brought the life expectancy back to what you would see in the 1930s. So this is, of course, a very dramatic impact on the overall health of the public in those countries. This is also a very striking figure. And once again, this comes back to the issue of women, girls, HIV and AIDS. And what we're looking at is the prevalence of HIV among teenagers in Kisumu, Kenya, by age. And we're looking at boys in blue and girls in pink. And what you can see is that roughly at age 15, only 15 years of age, virtually no boys are infected with HIV, but already at age 15, 8.3% of 15-year-old girls are HIV infected. Age 16, it jumps to 18% of girls, still no boys. Boys emerge as a group with seroprevalence by age 17. By then, we're up to almost 30% of 17-year-old girls. This dip is probably just based on the size of the study. What we can see is that by age 19, a third of young women in Kisumu that go to health clinics are HIV infected. This is an interesting question. Imagine if this was true of your classes. Imagine if a third of the young women in your classes were HIV infected. How would that affect what life would be like in your high schools? And the interesting question is, why is this the case? And why this dramatic difference in between girls and boys? Well, what is interesting in Kisumu is that it's in fact not uncommon for older men to sponsor girls in high school, and in fact to buy their textbooks, to buy their school uniforms in exchange for sex. And so what you see here is direct evidence of, in fact, the transmission of HIV from an older male population specifically to young girls in high school in this town in Kenya. And there you have a situation where a social structure and a very unfortunate sort of a situation driven, of course, by poverty is resulting in a seroprevalence in an age group that I think anyone would find appalling, and which ultimately will have a very dramatic impact on this society.
I have some friends who live in Cameroon at the moment. The father is a doctor, and they have teenage children with them. And when they first got to this village, they're in
title of the store within South Berkeley. Because they thought that that was such a compliment
Yes.
to ask for their daughter.
Yes, so there are really sort of serious cultural issues and structural issues that have to be kept in mind in terms of how the epidemic is unfolding. Sort of another very interesting example, which I'll touch on briefly. Don't think that Africa is the only region that is at very serious risk. As I said, the former Soviet bloc countries are now showing rates of new infection that are greater than 50%, not just in women, but overall. The number of new infections, for example, in the Ukraine, I believe it's over 65% higher in the course of 1 year than the previous year. So what you're seeing is that in many countries, the epidemic is really just taking off. And unfortunately, we're missing an opportunity to, if you will, nip those epidemics in the bud. A very interesting case is India. The World Health Organization predicts that unless something is done in India very seriously by the government in terms of AIDS education, by the year 2025, India alone will have more individuals with HIV than the current global total. They will be projected to have over 40 million HIV infections in India alone. Because of course, India has a population of over 1 billion. So what this means, of course, is that India sort of represents a real tinderbox for the disease. So that as serious as things may look, this epidemic has not really taken off in key parts of the world. So we haven't seen it at its worst yet, and that's something to keep in mind. And another very interesting thing about India, which you might be interested in knowing, is that in most countries, this disease begins in urban centers, and you slowly start to see it appear in small towns, small villages. India is quite different. The seroprevalence in small villages is nearly as high as what you'd see in Bombay or Madras. And here is a very interesting case of where sort of an economic structure facilitates the spread of the disease. In India, more than half of the goods are transported from southern ports to every little town in the country by truck. It's a country that ships not by train or by air, but they ship by lorry trucks. There is a population of over 1 million truckers that ship goods across all of India, from Bombay to basically every little hamlet throughout the entire country. Based on truckers that come into health clinics for treatment for STDs, at this point, greater than 80% of truckers in India are HIV positive. Furthermore, it is tradition that as soon as a trucker reaches his destination, the first thing he does is go to a sex worker, go to a prostitute in that town. So on average, a trucker in India has more than 300 different sexual partners per year, primarily based on interactions with sex workers. Why is that the case? Well, what it turns out is that for several decades now, the mythology in India among the trucking industry has been that when you drive a lorry truck, you absorb unhealthy heat from the engine, and that you must discharge that heat when you arrive at your destination, or you will go sterile. And the way to discharge the heat is through sexual intercourse. So here we see a remarkable synergy and a tragic synergy of an economic business structure with a mythology, with an urban mythology that results in the most alarming vector system for a virus that's sexually transmitted to go from urban centers to every little town throughout the entire country. So that's why India is another region that is of tremendous concern. But in any case, clearly the epidemic is not under control. And so the question that we face is, what is it about this virus that makes it so difficult for us to handle? One thing you should keep in mind, HIV is the most researched pathogen in human history. Each year, there are more than 16 to 18,000 primary literature research articles on this virus alone. We have known about this virus, we've had our hands on this virus basically since 1982. So over 2 decades of research, and A, we're far from a cure. B, we're far from a vaccine for this virus. And believe it or not, C, we still don't fully understand how HIV infection results in the full spectrum of disease that we understand as acquired immunodeficiency syndrome. So how is it that this pathogen can be so pernicious and so amazing? Just how complex is this pathogen? And so what I'd like to spend sort of the remainder of my time is talking about 2 things that this pathogen does that makes it truly remarkable as an adversary for us as humans. For one, there's almost no other pathogen that co-opts our defensive systems for its own pernicious means like HIV. So co-option is a critical issue. Also, through this process of co-opting normal sort of cellular and host immune system responses, it results in a sort of ironic twist on the destruction of the immune system. It does both of these things by a remarkable range of activities. I will touch on just a few of them this evening, but it'll give you some flavor of how this virus works. So what you're seeing here is sort of a colorized transmission electron micrograph of new virions that are budding from the surface of an infected cell. So this is an infected T helper cell, and there you see the new virions. And one might think that for this pathogen to be so deadly and so complex, it must be a very complicated virus. In fact, it is not. Here we see the structure of HIV. And what we can see is that structurally it is not that different at all than most of the envelope viruses that we can think of. So that if you look at HIV, like most viruses that have a phospholipid bilayer envelope derived from the cell that they bud from, you can really think of it in terms of 3 critical components, or layers, if you will, of organization. The first layer is the outer envelope or membrane of the virus. As one can see from this budding event, that envelope is actually derived from the host cell membrane, from the membrane of the infected cell. So that in fact, the infected cell actually contributes a patch of its membrane to form the envelope of the virus. And in the case of HIV, embedded in the envelope of the virus are 2 very critical proteins. One called GP120, glycoprotein 120, and another one called glycoprotein 41. As we'll see shortly, GP120 is in fact the key that unlocks the cell for HIV. And that viral protein is responsible for the specific docking to target cells which can be infected by the virus. GP41, on the other hand, actually allows this membrane to fuse with the membrane of a cell that the virus has docked to. And in fact, the mechanism of that fusion was actually solved here at Harvard and has given us sort of a new handle for possibly blocking infection by HIV. And if I have time, I'll talk about that. If we move further into the virus, we have a second layer of structure. This is called the viral matrix. And it's a complex of proteins that really give HIV a very stable structure. And this complex of proteins also allow the virus to bud from the infected cell. So a very important role. And then inside, in sort of the innermost layer of the virus, you have the core. So you have this complex of viral proteins, and housed in that core are two identical strands of RNA. HIV is an RNA-based virus. It is a retrovirus, and it has two copies of its RNA genome. And included in this core are, of course, the enzymes, which, as we'll see in a couple minutes, have a very important role in the replication cycle of the virus. And we also have some critical proteins in the core that, believe it or not, interact with host cell proteins to facilitate steps in the viral replication cycle. And we'll touch on some of those as well. So as you can see, not an overly complicated structure. And the genome that we're talking about is 9.8 kilobases in length. 9,800 bases, not particularly big. And in those 9,800 bases, you have 9 genes. That's all HIV has, is 9 genes. Nevertheless, those 9 genes can do a lot for the virus. And maybe we'll have a chance to talk about this tonight. There is almost no such thing as an HIV gene that has one function. Nearly everything in this virus is multifunctional. And I'll touch on some of those multiple functions as we go through the replication cycle. So, how does the virus replicate? Well, quite simply what happens is that HIV, in most cases, needs to bind to 2 critical host cell receptors. So, if you will, the canonical target for HIV are cells that carry the CD4 cell surface molecule. And as you probably know by now, CD4 is found on T helper cells, those critical cells that help sustain both the cell-mediated and the antibody-mediated immune responses. And CD4 is also found on macrophages as well. So that is, in fact, the primary receptor for HIV.
Does HIV attack a broad range of cells or only helper cells and macrophages?
That's an interesting question. For over a decade, it was thought that HIV only attacked T helper cells and macrophages via CD4. And in fact, for almost 15 years, it was thought that CD4 was the only receptor that HIV needed. In fact, in 1996, it was discovered that HIV critically requires, in many cases, another cell surface receptor called the co-receptor. And so as you can see, as the years go by, even the simplest of ideas with this virus, in fact, has to be revised as new discoveries are made. So in fact, in most cases, both CD4 and the co-receptor are required. The combination of those 2 receptors and how they interact with GP120 and GP41 allow the fusion event to take place. Once fusion takes place, the RNA genome is reverse transcribed into a double-stranded DNA copy, or viral cDNA, by viral reverse transcriptase. This viral cDNA is integrated into the human genome. And so one interesting thing to know is that in an infected cell, the HIV genome in the form of the provirus is indistinguishable from your own genome.
Is it going in only one place or anywhere all over?
So the other question is, so does it go in one site or is it randomly integrated? For over 10 years, it was thought that integration was entirely random. What we now know is that that's not true, that the process of integration is semi-random. And now here's an interesting question I have for you. So you're the virus. You need to get this viral DNA somehow fully integrated into the genome. You have a viral enzyme called integrase that comes along with the virus that actually catalyzes that process. There are certain hotspots in the human genome for integration to take place. Where do you think those hotspots might be? Where would you just predict those hotspots to be? Any thoughts?
If,
Shouldn't it be in repeating sequences that are found throughout the genome?
Okay, so one response is, could they be in repetitive sequences? And in fact, that is true. There are certain sets of highly repetitive DNA sequences that are more amenable to integration. But there's sort of another issue as well that's independent of the sequence of the DNA.
Is it maybe where the strands tend to break a lot anyway and have to be repaired, like maybe those thymine dimer things?
So another thought is it where DNA tends to fragment a lot, or where the process of DNA repair is taking place? Actually, the way that the HIV enzyme integrase works, it doesn't require a break. But you're touching on the critical issue in that those sites are also characterized by something else.
I'm just guessing. MHC, perhaps?
No, there's no involvement of the MHC region in the genome in this. But for example, sites where DNA repair is actively occurring, among other things, are characterized by being decondensed. Because if you think of it, as you know, DNA is very tightly packed with histones. If you want to plug in another piece of DNA, the more naked the DNA is in that part of the genome, the better it is for you. So HIV, preferentially, naturally enough, will integrate at sites in the genome where the DNA is unpacked or exposed. Now, here's something that's also very interesting. What parts of the genome, other than those that are being repaired, tend to be exposed or unpacked? What's that? The ones that are being actively transcribed. So lo and behold, by the simple mechanical need to integrate in a region of the genome that's somewhat naked or unpacked, that provirus lands in the most transcriptionally active regions of the genome. What does that allow the virus to do? Well, quite simply, it takes advantage of the transcription that's already occurring at those sites. So that transcription factors like NF-kappa B, not only will they transcribe the genes that they're supposed to be transcribing, they'll transcribe the provirus as well. Very simple, very elegant. And this is a theme for this virus of very simple but remarkably elegant mechanisms. So, based on landing in the right place, the provirus is transcribed, viral proteins are produced. Two critical proteins are TAT and REV. What's interesting about TAT? TAT actually increases, it gets back into the nucleus, and it increases proviral transcription specifically by over 1,000-fold. So HIV expresses its own transcription activator that enhances transcription of itself by over 1,000-fold. You get the production of many other viral proteins, including structural components, that then allow the formation and the budding of new virions. Now, I just can't resist. Here's another example of something very elegant. Let's look at TAT. For over 15 years, TAT was a focus of major research because A, TAT is essential for HIV. If you have a mutant form of HIV that lacks TAT, it will not replicate at a high enough level in terms of transcription to drive the production of significant amounts of virus. So TAT is absolutely essential. For over 15 years, TAT was thought of as this remarkably powerful transcriptional activator. In the course of those 15 to 20 years of research on TAT, it was discovered that TAT works as a transcription factor in a way that is completely different from any other transcription factor known. And if I had time tonight, I'd share with you how that works, but I don't have time. So it's very, very interesting, and if you're interested in reading about it, I would recommend that you pursue it. There's something else about TAT, though, that I'd like to tell you. TAT gets back into the nucleus. We know that. any large protein or any protein that needs to get into the nucleus must do so by a very specific process of nuclear import. That requires a number of proteins in the cell that are involved in the process of nuclear import, a whole class of proteins called karyoferrins. So for the longest time, it was thought that TAT somehow hooks up to the host cell machinery of nuclear import. That's how it gets back into the cell, and that's how it transcriptionally activates or enhances the transcription of the provirus. Roughly 5 to 6 years ago, it was discovered that TAT does not need anything to cross cell membranes. So TAT is a protein that by itself moves across freely, diffuses across membranes. And I'm sure that all of you have taught your students proteins can't get across membranes unless there's a channel or a transporter. There's no way for a protein to cross the hydrophobic core of a membrane by itself. TAT has solved that problem. And in fact, there is a 23 amino acid stretch of TAT that allows it to embed in the membrane and flip from one side to the other. So TAT freely crosses into the nucleus. So that's kind of neat, don't you think? That's kind of interesting. But not only does TAT cross into the nucleus, in this infected cell, TAT crosses the plasma membrane and enters the bloodstream. So that infected cells shed TAT into the bloodstream. Now here we have an interesting possibility that has now been proven to be a reality. What is the consequence of that? Well, here you have your bloodstream. You have infected cells floating around. They're secreting, they're shedding TAT by diffusion into the bloodstream. That extracellular TAT, as it's called, will go into neighboring cells, which are also infected, but it'll go into neighboring cells that are uninfected. TAT will go into an uninfected cell and will cross into the nucleus. What is the consequence of that? When that uninfected cell becomes infected, this rate-limiting step is gone. As soon as the provirus becomes integrated, the nucleus is already loaded with TAT, and you get maximal HIV transcription immediately. So this is one means by which HIV actually primes uninfected cells for infection and viral replication, just based on a remarkable molecular characteristic of this transcription factor. Now, isn't that elegant? Isn't that amazing? So, these things are coming out nearly every other year. There's a new major discovery that has this degree of elegance. You had a question? No, go ahead.
At what point is the body sort of find out that being invaded by TAT, and is interferon being produced greater quantities to sort of overcome this process to some extent?
Okay, so the question is, when does the body realize or recognize, when does the immune system recognize that HIV is there and this is going on, and when and what happens, for example, with interferons? One thing to keep in mind is that for many years it was thought that HIV evaded the immune system. That the reason why there was a long period where you seemed healthy was because you didn't know you were infected. That is entirely sort of incorrect. Your immune system sees HIV right away. Right away. And in fact, the war with HIV begins within the first 48 hours of infection. The immune system battles HIV for more than 8 to 10 years. So in fact, in individuals that are infected, there's a pitched battle that goes on during that entire period. So the immune system sees it right away. What happens is that due to things like this priming of cells, of uninfected cells, for high-level replication, so much virus is produced, and we'll talk about that shortly, that your immune system just can't handle it. And that ultimately is what happens. What happens with interferons? HIV infection over time actually turns down the production of interferons. So the virus actually destroys or reduces the ability of the immune system to produce those things that are most effective at inhibiting the virus. And we'll see a couple of examples. Your question?
This protein TAT seems very scary and very unique. Are there other proteins in other genomes that are similar to it?
Well, in a growing class of viral pathogens, we are discovering some proteins that can do the same thing. And the whole class of them are being described as penetratins. So this is a new class of proteins. What is interesting about this is that not only are these interesting for understanding on pathogenesis, but because we now know the 23 amino acids that allow this to happen, a number of biotech companies are hooking up those 23 amino acids from TAT to other proteins. And in fact, you can hook up the penetratin domain to a therapeutic protein, and lo and behold, you can get that protein into a cell. So one positive of this is that discovering this phenomenon may actually allow us to create protein therapies for the first time that don't require some complicated transport mechanism for them to work.
The transcription, anything else, or just the HIV stuff?
I'm glad you asked that question. Does TAT affect the transcription of anything else? Well, for a long time we thought, well, TAT affected the provirus, that's bad enough. In the last year, it has been discovered TAT also increases the transcription of the co-receptor. So not only does it prime uninfected cells by making them ready to replicate HIV, it actually makes them more susceptible to HIV by upregulating the amount of co-receptor on the cell surface. Nasty, but quite amazing. Okay, one more question.
I know anything about, like, TAT as a protein. It has these 23 amino acids, and they're mainly hydrophobic, and how big is the rest of the protein?
TAT is not very big. It's only 86 or 88 amino acids long. So it only has 3 major functional domains. But what's interesting is each of those domains we're now realizing are multifunctional. And so that's part of the issue, is that each of these viral proteins are truly multifunctional. And for those of you that are interested in evolution, you might be wondering, how did this virus actually get to be like this? And if we have time, we'll talk about that at the end. Okay, one more question and then.
Does TAT have any relationship with oncogenes or tumor suppressor genes?
No. So the question is, yes, is there any relationship to growth control genes like oncogenes and tumor suppressors? No. One thing I should say, though, is those hotspots for integration, those are frequently oncogenic sites. Those are positive growth regulators. So in fact, what's also interesting is that the integration of the provirus frequently results in the hyperactivation of cell division. And in the process of hyperactivating cell division, the cell produces a lot of components that the virus then uses as a result. So everything fits together very beautifully. And I guess what this whole thing implies is that this virus has been dealing with a primate immune system for a very long time. And that's true. So, when the virus infects you, what we can see is that there is this immediate and enormous spike of viral replication in the body. So that, as I said before, it's not as if your immune system doesn't see the virus, it sees it right away. And as soon as you have this tremendous spike of viral replication in the course of 3 to 4 weeks, during that same time, if you look at the number of T helper cells, you lose roughly half of your T helper cells within that same period. So believe me, the battle that occurs, it's like those battle scenes in Lord of the Rings. It's truly an epic battle from the very beginning between the virus and your immune system. But remarkably, your immune system is very powerful. And within the course of 6 to 9 weeks, your immune system actually gains a foothold against the virus and brings the viremia down to relatively low levels in the bloodstream. So that what happens is there's a period of clinical latency where the amount of virus in the bloodstream doesn't seem that high. But what's happening during that time is that the virus is actually being seeded to lymphoid organs throughout the body. And so there's a process of colonization of your body by the virus that occurs over 1 to 6 or 7 years. During this period of clinical latency, the virus is far from dormant, and we'll see what the numbers look like shortly. And what happens is eventually your immune system essentially cannot deal with it anymore. Your immune system collapses, viral levels start to rise again, and you get the onset of opportunistic infections, the onset of constitutional symptoms, and ultimately, if you're not treated with antiretroviral therapy, you see death on average after 10 to 12 years. So this is the course of the disease based on viremia and the number of CD4 cells in the bloodstream over the course of 10 to 12 years. So
Question about the clinical latency. At this point, if drugs are administered, cocktail form, how is that going to change? I mean, obviously it's going to help out, but how is it?
Well, with gold standard drugs with combination therapy, the clinical latency is extended by roughly 3 to 4 years. So I think we're going to take a break now. But when we come back, what we'll look at is just how serious is the battle that's occurring during that period of clinical latency. And then we'll look at some of the molecular things that HIV does to ultimately gain the upper hand in that fight.
We're just going to take a 5-minute break so that you can run to the restroom if you need to. There are restrooms. There's one right after this. Before we do, I just want to say a word or two about our brand new website, which went up last night. And I hope if you haven't had a chance to check it out, that you will. In fact, I require you to check it out because on the last page of this evaluation, you will see I have written a little web quest for you. I need your feedback. Nothing's perfect the first time we try it. And so I want to know whether you have trouble finding anything. Okay? So please make sure you take a look at this and try to find little items that I've listed here. And any kind of difficulty, if you encounter any difficulty, please write it down on this page and let me know. Okay? A couple of you have asked me a couple of questions about the website. One pertains to when are these immunology lectures going to appear? And the answer is on December 15th. Okay? The immunology lectures should be there and accessible to you in QuickTime. And the PowerPoint presentations, I believe, should accompany them. Okay? After December 15th, if you have difficulty, please let us know. Okay? Another one or two of you asked me about the animations. We are going to, by December 15th, put up the drafts of the immunology animations. They will still be, they're definitely drafts. We're going to be working on them. And also something you should know about the cancer animations. A couple of you have said, hey, wait a minute, I just noticed that the animation I used last year isn't there anymore. There's one animation in particular from the cancer program that I am reworking. And actually, in response to some of your requests, I'm going to put that draft back up, but I'm going to write underneath it that it is indeed a draft. It used to be called the cell cycle. I've taken the cell cycle cancer animation, I've divided it in half. The actual cell cycle portion is up there on the website. The portion, for those of you who are familiar with it, with a petri dish of cells, when you choose the cells and follow a biochemical pathway for that cell, that's the part that's under revision. That's been separated. I'll call it pathways, but that draft will be there for you. Okay? So if there are any other questions about the website, please let me know. But thank you.
Where were we? Oh, so so as I mentioned before, there's really been a change in our view of what happens during that period of clinical latency. So that for some time it was thought that perhaps the virus was dormant. But in fact, what we know is that there's a pitched battle that's going on in between the two sort of sides, if you will. And that in fact there are surprisingly high rates of HIV replication and CD4 cell turnover during the course of clinical latency in HIV positive patients. So what that means is that not only are enormous quantities, are numbers of virus being generated, but that also large numbers of cells are dying and being replaced, regenerated from the immune system. So that what's interesting is that what we know about HIV and how it interacts with the immune system has in fact changed our view of how resilient the immune system is when it comes to regenerating certain key cells, in particular T helper cells. So that by current estimates, roughly 100 billion new HIV particles are generated each and every day in the body of an infected individual, even one that seems perfectly healthy. Over 100 billion. During that same day, 1 to 2 billion CD4 cells are killed and replaced during that day. So what this means is that enormous stress is placed on thymic centers, on thymocytes, on the thymus, because of course it must produce all of these new T helper cells that ultimately replace the billions which are destroyed by the virus each day. So what that means is that even in an individual that seems healthy, where they're in the process, in the stage of clinical latency, there is an enormous battle that's taking place.
Your thymus gets smaller and smaller the older you get. So there's a point when you can't replace it.
Right, so the question is that your thymus shrinks with age, that's quite true. And that's why, for example, even with individuals that have successfully controlled the HIV sort of infection with therapy, as they grow older, it becomes increasingly difficult. And it's because the thymus does shrink over time. And in fact, one therapy is trying to develop some means of preventing that from happening. So clearly the battle is being waged. And one of the problems that the immune system faces during this battle is that, unfortunately, the very comfortable idea that HIV uses CD4 and co-receptors to infect cells is not 100% true in all cases. So that what we now know is that HIV actually can use alternative receptors. And this is a very troublesome issue, because if you're trying to figure out how a pathogen works, you want to be able to understand the host range of that pathogen. You want to know what is necessary and what identifies a target cell that allows that pathogen to infect that cell. Basically, for well over a decade, for almost 20 years actually, it was thought that CD4 is the critical thing. And in fact, it's that handshake between GP120 and CD4 that allows infection to take place. But there was always an experimental observation that was troubling in that regard. And this was the very simple experiment that led to this observation. If you take mouse cells that express human CD4, if you transfect mouse cells to express human CD4, in theory, if CD4 is the only player, those cells should be infectable by HIV. That is not the case. While if you fuse those mouse cells that are expressing human CD4 with human fibroblasts that don't express human CD4, lo and behold, the hybrid is infectable. So that there are other proteins that are involved in human cells. And of course, some of those critical other proteins are the co-receptors. So that's straightforward. Nevertheless, though, even from those experiments, co-receptors always require CD4. So a co-receptor like CCR5 by itself will not allow HIV infection. But interestingly enough, there are cells that appear to be susceptible to HIV that don't require CD4 at all. These CD4 so-called independent cells, or CD4 minus cells that can be infected by HIV. And these cells include brain astrocytes, no CD4, but HIV can infect them. Bowel and renal epithelia, very important target for HIV. No CD4 whatsoever, but are nevertheless still infectable by HIV. And certain dental pulp fibroblasts don't carry CD4, but are still infectable by HIV. So, what are these alternative receptors, and how does HIV use them? And there's one alternative receptor in particular that I'll spend a few minutes talking about. A, because it's very important in sexual transmission, which accounts for more than 3/4 of new new infections globally. And B, because it actually involves a terrible misuse of the immune system for the purposes of the virus. And this alternative receptor is what's called the Fc receptor. And the Fc receptor plays a very fundamental role in several key immune system processes, because the Fc receptor is actually a key part of the immune system. So, what is the Fc receptor? So before I tell you that, I have to tell you what Fc is. And have you encountered this in the talk so far? Okay. So before I tell you what the receptor is, what is Fc? Well, I know you've seen this, the antibody molecule. And of course, well, you probably know, you have 2 heavy chains and you have 2 light chains. These are the variable regions, which is where antigen binding takes place. Highly variable from one class of antibody molecule to another. This, on the other hand, composed exclusively of heavy chain sequences, is the so-called constant region. So what the Fc receptor actually does is that it binds to the Fc or constant portion or fragment of the antibody molecule. So Fc receptors bind to the Fc portion of antibody molecules. So, what does this have to do with HIV? Well, Fc receptors are found on a variety of cells, including B cells, macrophages, and neutrophils, and it is also found on bowel epithelia. In fact, bowel epithelia are highly enriched in Fc receptors. What does it do in B cells? Well, its normal function in B cells, and as you know, B cells secrete antibodies, is that the Fc receptor on B cells will bind to excess antibody in the bloodstream in late-stage immune responses. And this actually induces the B cell to cut antibody production. So it plays a critical role in the normal feedback or negative feedback process in late-stage immune responses. It also has a very important role, for example, in phagocytes like macrophages, in that it enables the recognition of antibody-coated bacteria and allows for the phagocytosis, or the engulfing of those bacteria. So this is a process you have probably perhaps mentioned to some of your classes called opsonization. So that what happens quite simply is that you have a bacterium that is coated with antibody molecules. Of course, the Fc portion is facing out. That Fc portion binds to Fc receptors on the body of a macrophage or neutrophil. This allows the zippering up of the membrane around the bacterium. The bacterium is engulfed by the macrophage and broken down in the endosome. You have seen this before, and you may have taught this to some of your classes. Now, where does HIV come into this? Well, let's look at this very nice diagram and replace the bacterium with HIV. When HIV is coated with anti-HIV antibodies, those antibodies will bind to Fc receptors. The virus will be phagocytosed by the cell. What HIV does, however, is that unlike this poor hapless bacterium that has no choice but to remain in the endosome until it becomes a lysosome and it's broken down, what HIV does when it's taken up in the endosome, it fuses with the endosome. And by fusing with the endosome, the viral core is injected into the cell and you get infection. So what HIV literally does, it takes advantage of what is a very nice antibody-mediated mechanism for destroying pathogens, and it uses it to get into the cell. And that is exactly what happens with bowel epithelia, for example. And in fact, that also happens with cervical, cervical epithelia as well, which are highly enriched in the Fc receptor. So here we see one very nice example of how HIV literally takes a very normal mechanism of the immune response, and it takes antibodies, those warriors of the humoral immune response, and it uses them, it co-opts them and uses them to infect cells. Very pernicious, very nasty mechanism. Question?
If a virus, it can be coated with the antibodies, then bound to a B cell,
Right.
would that then tell the B cell to quit making antibodies and thereby slow down that immune response against the virus?
That's a very interesting question. Would an antibody-coated virus signal to a B cell to stop antibody production? At this point, there's no evidence for that. It appears as if in normal function, multiple Fc receptors must be activated by antibody within a certain time period. And so just a virus binding or even several viruses binding would not allow that to happen. And it's quite interesting. HIV actually likes antibodies. It loves them because it can utilize antibodies not just in this way, but via other mechanisms as well. And we'll talk about that in a few minutes. So, obviously antibodies are very important. It's also well demonstrated that antibodies can neutralize HIV. So there are certain antibodies that can bind to HIV, strip off the GP120, and neutralize the virus. So almost from the time the virus was discovered, scores of labs have spent a great deal of time trying to identify what are called the neutralization sort of epitopes on HIV. Or quite simply, what are those epitopes or sites on HIV that when an antibody binds to them, the virus is neutralized. The so-called neutralization epitopes. Now these are important, in particular for folks thinking about a vaccine. Because if you can find a neutralization epitope on HIV, you can take that epitope and use it as a vaccine. Because if you introduce it into individuals, they will produce antibodies against that epitope such that if HIV comes in, those antibodies will bind and neutralize the virus. Very clean, very simple, Sounds nice. It's complicated. But still, let's look at this sort of issue of neutralization epitopes. What has been found is that a prime target for neutralization is in fact the GP120 viral protein. And that if you compare the GP120 sequence across different strains of HIV, there are regions that are variable and regions that are constant. And there are roughly 5 of these regions each. 5 variable regions and 5 constant regions. So that if you look at the clamshell structure of GP120, you have, for example, variable regions 1 and 2 that are highly exposed in that loop. You have variable region 3 that's highly exposed. And you have, for example, an alpha helix that is a constant region called alpha 1. So there's sort of a variety of regions which are variable versus constant in GP120. What has been found is that on the viral envelope, there are 5 major neutralization sites that have been identified so far after years of work. 4 of these sites are on GP120, and there's another site that's on the other viral envelope protein, GP41. So there's been some success, seemingly, at discovering or identifying major neutralization sites, in particular on the GP120 structure. Now, if we look at these GP120 neutralization epitopes, the major one is in variable loop 3, the V3 loop. And in fact, neutralization often occurs at the central portion of the V3 loop, which is frequently called the principal neutralization domain of the virus. So the PND in V3 is the primary neutralization site for antibodies that seem to neutralize HIV. Sounds good. We could use PND as a vaccine. What also appears to be true is that V3 neutralization appears to involve a post-binding step of HIV entry. And what we now know is that V3 actually interacts with the co-receptor. So that in fact the neutralization mechanism may involve the blocking of the subsequent co-receptor interaction step. Very nice. So we may have a vaccine on our hands. Well, as you know from the FC receptor story, antibodies don't just neutralize. In the case of HIV, they're antibodies that enhance the virus, or in fact enhance the infectivity of the virus. These so-called enhancing antibodies. So at the same time that labs wanted to identify neutralization epitopes to create vaccines, labs also wanted to identify enhancing epitopes to avoid those regions. Because can you imagine how terrible it would be to vaccinate with an epitope that when the virus came in, instead of neutralizing it, would enhance the infectivity of the virus? That would be terrible. So, this process of enhancement, as you recall, is via the FC receptor. That's just actually one sort of possibility. And what had been found is that in many patients, if you sample blood from patients, they show this mixture of both antibodies that will enhance and antibodies that will neutralize the virus. And that in fact, if you take blood serum from patients that is generally neutralizing in character and you dilute it, it actually becomes enhancing when you test it on cells in culture that are exposed to virus. So how can that be? Well, what we think is the case is that blood serum from patients generally contains a larger number of low affinity binding enhancing antibodies. and there's a smaller or lower concentration of high affinity binding neutralizing antibodies. So that when you have blood serum at full strength and you test it on cells in culture that are exposed to virus, there's still enough neutralizing antibodies to result in net neutralization. But if you dilute the serum back and then add it, you don't have enough neutralizing antibodies for net neutralization, but you still have enough enhancing antibodies to support the enhancement of infection. So that the simple idea is that in patients, there appears to be this balance in between neutralizing and enhancing antibodies. So clearly a problem that has to be kept in mind, but let's forge ahead and identify those enhancing epitopes so that we can avoid them at all costs. Sounds like a plan, don't you think? So, big effort to identify those viral epitopes that are relevant to antibody-dependent enhancement of infection, or ADE, like the Fc receptor-mediated mechanism. A lot of effort went into looking for these epitopes. What was found was that so far there seem to be fewer of these epitopes identified than what we know for neutralization. There seem to be 3 critical ones which have been identified so far. Interestingly enough, they still focus on GP120 and GP41. So what are these enhancing epitopes? Lo and behold, the V3 loop has sites shown to play a role in Fc-mediated antibody-dependent enhancement. So the very same domain that might have a role in neutralization might also have a role in enhancement. So how is that possible? How is it that the same regions involved in neutralization can be involved in enhancement? Well, clearly what we now know is that it depends on the strain of HIV that's involved. So that if one antibody binds to strain X, it will neutralize it. If that same antibody binds to strain Y, it will enhance it. And now you see we have a very serious problem in terms of how antibodies interact with the virus. And so there's a very simple experiment that strikingly underscores this problem. So, what I just implied was experimentally demonstrated. You have antibodies that enhance one HIV strain and neutralize another. How do we explore this process? Well, we'll use both human and murine monoclonal antibodies. And as you know, monoclonal antibodies bind to one epitope. So these are well-characterized monoclonal antibodies that only bind to one place on HIV. So let's test one of these monoclonals, and in particular a monoclonal that binds to a small epitope of 6 amino acids in the V3 loop. So we have this antibody, we know where it binds. It binds to these 6 amino acids in the V3 loop. Lo and behold, this same antibody has 3 entirely different responses when applied to 3 different strains of HIV. What a nightmare. If you apply it to the SF2 strain, it neutralizes the virus. The virus cannot infect cells. Sounds good? If you apply the same antibody to SF128A, a different HIV strain, it enhances the efficiency of infection, and you see increased viral infection. If you apply to SF162, there is no effect. So this is described as resistance. The antibody has no effect on the virus at all. It doesn't block infection. It doesn't enhance infection. So we have one antibody, 3 strains, 3 responses. We know where the antibody binds. So of course, what does this allow you to do? It allows you to look at that binding site and see, well, is that binding site different in any way? What are the sequences at the epitope itself? And lo and behold, here's the amino acid sequence of the SF2 epitope, the one that supports neutralization. Here's the sequence at the 128A epitope, the one that supports enhancement. And here's the sequence at the SF162 epitope, the epitope that supports nothing, where there's no effect of the antibody binding at all. And as a man on a galloping horse would notice, the difference is 1 amino acid. So there's only 1 amino acid difference in the epitope. So that this variation results in this spectrum of responses to exactly the same antibody. So in the case of SF2, when there's a tyrosine, a hydrophobic amino acid, you see neutralization. When that amino acid changes to a hydrophilic amino acid like asparagine or threonine, you switch to either enhancement or to no effect at all. In terms of binding affinity, with SF2 it binds most tightly. SF128A has intermediate binding affinity. SF162 has the weakest binding affinity. So in the case of HIV and its interaction with antibodies, it's a little bit like, do I have the right fairy tale? Like Goldilocks and the porridge. So that what you have is a situation where if it binds very tightly, you get neutralization. If it binds very weakly, you get nothing. But if it binds just tightly enough to connect the virus, let's say with a macrophage, and allow the engulfment, then you will see the antibody-dependent enhancement. And the problem is, can you generate an antibody where you can predict what the response will be? So to explore this a little bit further, and I just told you all that, to explore this a little bit further, what would happen if we mutagenized the actual epitope? Can we convert one response into another response? So if we change the single amino acid, that would alter the SF2 sequence into the SF128A sequence. Would we go from a neutralizable strain to an enhanceable strain? If we could do that, we'd know that it's just the epitope. That didn't happen. It changed SF2 to resistant, not to an enhanceable strain. So clearly the epitope is not everything. And in fact, the implication is that it's not just the sequence where the antibody actually binds, it's the presentation of that sequence in the context of the 3-dimensional structure of GP120. So that in fact, if you change 3 amino acids in another part of the V3 loop of SF162, which was the resistant strain of HIV, you go from resistant to neutralizable. The epitope is exactly the same, but you change 3 amino acids elsewhere it changes the presentation of the epitope, it changes how the antibody binds to it. So as you can see, it's getting even more complicated in terms of predicting what an antibody will do when it binds to the virus. So that clearly a single epitope is not the sole determinant of the antibody effect as far as HIV is concerned. And this raises a serious clinical warning. We cannot hope to identify regions of the virus that will only produce a neutralizing antibody response. Obviously, this would be needed for sensible and safe immunization, so that ultimately an antibody response that is neutralizing for one HIV strain could enhance the infectivity of another. And unfortunately, with every vaccine approach being explored, any vaccine approach that creates an antibody response runs the risk of creating one that could potentially enhance the infectivity of an HIV strain that that individual is exposed to. So this just begins to scratch the surface of the complexity of why it's so difficult to handle this virus, because it literally uses and co-opts the immune system for its own means.
Is there a difference in the strains of their infectious rates through the CD4 co-receptor pathway?
Okay, so is there a difference from one strain to another when it comes to the infection route via CD4 and the co-receptor? Yes, there is. And this is a very interesting thing that I didn't touch on. There are in fact 13 different co-receptors. HIV can use one of 13. The co-receptor that's preferred depends on the strain. So let's talk about the simplest sort of distinction. Macrophages carry a co-receptor called CCR5 primarily. T helper cells carry a co-receptor called CXCR4. Certain strains prefer CCR5 and thus have a tropism, a preference for macrophages, and are described as macrophage tropic. And that is the basis for macrophage tropism. Other strains of HIV have a preference for T helper cells and have a preference for CXCR4, so that co-receptor defines T helper cell tropism. This was discovered only 4 years ago, basically. What's important is that 9 out of 10, in fact probably 9.5 out of 10 HIV infections via sexual transmission, which is most of the HIV infections, are of macrophage tropic strains. So CCR5 is the critical co-receptor for sexual transmission, because it primarily allows infection of macrophages. Why is that important? Well, the genital tract, in particular the vaginal canal, is highly enriched in macrophages. So in terms of penile vaginal sexual transmission, macrophages are the primary targets that are in the right place at the right time. So macrophage tropism and that co-receptor tropism is critical for the initial transmission event by sexual transmission via heterosexual means. But there's another twist. Macrophages live with HIV infection much longer than T helper cells. A T helper cell will live on average for 2 to 4 days with HIV infection. It cranks out an enormous amount of virus and eventually dies after a couple days. A macrophage with HIV can live for 2 to 3 weeks. What you may not know as well is that macrophages are highly mobile. They go crawling all over the body. There's a subset of macrophages called dendritic cells, whose role in the immune system is to migrate throughout the tissue and look for pathogens. From the point of view of HIV, if it can hook up with a vector that lasts longer and travels all over the body, isn't that better for seeding the body initially with the virus? The answer is yes. So that's why macrophage tropism is so important, and that's why the preferential use of one co-receptor is so important in sexual transmission. I digress, but this is another very important sort of issue with HIV and another elegant example of how it really uses what the human immune system, what the primate immune system has to offer.
Can I follow up?
Yep.
The reason I was asking is I was wondering if the strains have apparently different infectivity based on this non-CD4.
Right.
I was wondering if that correlates at all with their infectious rates through CD4. So the ones that are enhanced by the FC receptor pathway might have a lower infectious rate through the classical, if you will, pathway of CD4.
So do the ones that are particularly amenable to FC receptor mediated infection, are they less efficient via the CD4 route? Not really. No. These alternative receptors generally function in addition to the CD4 route, which is partly why the colonization of tissues is such a problem with this virus. So it's not as if subsets of virus are restricted in tissue sort of range. They all have the CD4 range, plus subsets have these additional ranges as well. And what's interesting, though, is that over time, the strains of virus that emerge in the body over 5 years, 6 years of infection, they tend to be more amenable to the FC receptor mediated mechanism. So that ultimately the antibody response turns against you over time as HIV is concerned. And what is also remarkable, I'll just jump ahead a little bit because I don't think I'll get to this. Macrophages that are infected with HIV upregulate the secretion of cytokines that enhance the secretion of antibodies by B cells. So that came back to an earlier question, does HIV turn down the production of antibodies? Actually not. On the contrary, over years with the disease, HIV turns up antibody production because over time, as strains diversify in the body, HIV loves those antibodies because they in fact enhance its ability to infect any cell with an FC receptor. So as you can see, over time, HIV does all sorts of things to enhance the infection rate on uninfected cells. So that's a really remarkable feature. The virus uses antibodies to its maximal sort of means possible. I was going to tell you how HIV actually directly kills cells that are not infected. There's a remarkable process called the bystander effect, where HIV is able to destroy uninfected cells. So that here we have a virus that doesn't even need to infect the immune cell, it can kill it indirectly. And that's in particular true of T helper cells. So that HIV doesn't need to infect a T helper cell to kill it. By altering the expression profile of cytokines by macrophages, HIV can induce uninfected T helper cells to undergo programmed cell death. So that's yet another mechanism for reducing the number of T helper cells in the immune system. But I won't have time to talk about that. What I would like to talk about is how can HIV do all of this with the human immune system? It has clearly evolved a remarkably complex series of back and forth interactions with the immune system based on multifunctional proteins, so on and so forth, that implies that HIV has dealt with a primate immune system for a long time And that, in fact, is very true. So that HIV has been present in a particular species of chimpanzees for probably thousands of years. So the big question, where did HIV come from? HIV came from chimpanzees. And that in fact there's a particular species of chimpanzee called Pantroglodytes troglodytes that has probably been infected with HIV by current estimates for tens of thousands of years. And the interesting thing is, Pantroglodytes is perfectly healthy with HIV infection. Perfectly healthy. So this was a discovery that was made 2 years ago. And one can immediately understand why this is exciting. Because in Pantroglodytes, we have a primate that can live with HIV. If we are willing to let the HIV epidemic run unchecked in 10,000 years, we'll probably be in the same place. We'll probably lose on the order of 80% to 90% of the human population. The survivors will be the next Pantroglodytes, and will be able to live with this virus. Naturally, that is not an acceptable turn of events, obviously. But if we can understand how this primate, how that immune system in Pantroglodytes controls HIV, we'll understand how to beat this virus. So obviously, every Pantroglodytes has this back and forth with HIV that makes it a livable infection. And in fact, it's thought that the Pantroglodytes immune system is particularly robust based on its sort of back and forth kind of interaction over time with HIV. But there's a complication. Pantroglodytes is an endangered species. And this is in fact intimately linked with how the virus got into humans in the first place. In countries in Central Africa, like Cameroon, like Gabon, the construction of highways into the rainforest has allowed the interaction of species like Pantroglodytes with humans to a degree that has never been seen before. And it is thought that in fact Pantroglodytes entered humans off and on over the course of the last 30 years probably, or 40 years. But that with the lack of significant international travel, individuals that were infected with the virus died in small communities, and the disease never got anywhere. Now with highways going through the jungle, there's the transport of infected individuals from small villages in the jungle to urban centers, and we see the movement of the virus into the human population in the early 1970s, in the late 1970s, moving into the early 1980s. That same process is in fact driven by what's called the bushmeat trade. If you drive down the central highway that cuts through the rainforest in Gabon, you will see stalls that have a remarkable range of exotic animals that have been butchered for meat. In a society where a source of high quality protein is critically important and in short supply, the jungle represents a major source. So you will go past these stalls and there will be 8 to 10 Pantroglodytes that have been filleted and butchered and are available for sale. This has two consequences. A, the capture and butchering of the animals allows the transmission of the virus to humans. B, it's driving them to extinction. If Pantroglodytes goes extinct before we can understand how it controls HIV, we would have missed one of the most important opportunities for controlling this disease. But so that is of major importance. But the reason why I bring this up is because it really helps you understand that in fact HIV is highly evolved over tens of thousands of years to play an ongoing 10 to 20 year chess game with your immune system, where ultimately your immune system will be checkmated by HIV, unless you're a chimpanzee. So, on that note, we have some hope for understanding how this virus interacts with the immune system. But as you can see, I have just scratched the surface of the chess game that HIV plays with your immune system over the course of more than a decade. And ultimately, it is a chess game that your immune system will lose almost assuredly. In all likelihood, tens of thousands of years ago, the pan-troglodytes population was decimated. And probably 90 to 95% of the population was wiped out by this virus. The benefit for those that survived in the jungle is that those that survived had an immune system that is much more robust and in all likelihood able to regenerate cells in a much more robust fashion that gave them a selective advantage in the jungle, where you're not only dealing with this virus, you're dealing with other bacteria and other types of viral pathogens as well. So that ultimately, if we were to let it run its course with humanity, those that would be left would probably have an immune system that is much more robust. So the prediction is, we all know that the human immune system declines over time. That ultimately, once you pass the age of 40 to 50 to 60, your ability to regenerate new T cells is severely compromised by the atrophy of your thymus, among other things. The idea is that we would evolve as a species that would have an immune system that would last much longer and be much more regenerative, and that would be the consequence of our interaction with this virus. Of course, we cannot allow that kind of selection to ultimately happen for the human race. What is also important to note as well, and this is just sort of an interesting thing to think about. I didn't have time, unfortunately, to talk about some of the wonderful new therapeutic approaches which are being explored. The virus still develops resistance to all of these approaches. That has not been solved yet. One thing you should keep in mind is that a single individual that's infected with HIV represents sort of a population genetic study in and of themselves. You are infected primarily by one strain of virus. By the time 6 months has elapsed, you probably have on the order of 12 to 20 different strains. So the virus becomes an evolutionary case study in just your body alone. What is increasingly problematic is that as we have individuals that are on combination therapy passing on the virus, people are being infected now. At this point, roughly 30% of new infections in the US are with strains that are already resistant to combination therapy. So that's a little tweak that you have to keep in mind in terms of the new infections that are occurring and in terms of the utility of therapy. Another tweak to add on top of that is a life-saving therapy is only life-saving if you can get it. At this point, only roughly 3% of individuals that are HIV infected have any hope of a sustained treatment with combination therapy. So for all of those people in Africa, in India, in Southeast Asia, in Latin America, for example, combination therapy is not an issue. So that clinical course that I showed you is what they're looking at. They'll be dead in 10 years. And actually in Africa, it's more likely from the first time you're infected, you'll be dead in roughly 7 years, 6 to 7 years. So all of these wonderful therapies, unless we as a society make a concerted effort to make them available, even in their imperfect state, they're not an option for the majority of folks which are HIV infected. So in any case, on that point, there's hope, but there's still a very serious challenge facing humanity. So thank you for your time.